I'm Raghunathan, and I work in the chemical engineering division, but I am a systems biologist, and I like to call myself an emergent systems biologist because systems biology is all about understanding emergent properties, right? Do you all agree? You do. Okay, great. So what we'll do is today in the first half of my presence in this room, we will go more pedagogical, and this is what I'm going to talk to you about. So we are going to go through a journey of how do you build metabolic models from biochemical network reconstructions, and then how do you apply constraints-based modeling approaches to these networks. And so I love this title, From Genomes to Life. I've actually copied it from the Department of Energy in the US. They had a funding program called Genomes to Life where they actually sequenced genomes. I think through this pedagogical talk and through the next talk that I will give on my research, you'll probably understand more why I love this title, but we'll move on. So before I move on, I need to acknowledge that this pedagogical talk, parts of it have been adapted and modified from the Haugen lectures given by Bernard Paulson, and some of the metabolic engineering notes by Gregory Stephanopoulos. So let's start with why we need to build network reconstructions and metabolic models. So one example is infectious disease. Infections are a very significant problem in the world, and almost 20% of the population dies because of some form of infectious disease. The drug pipeline is drying up. The number of drugs that have been approved over the last several years by the FDA has been going down. Existing drugs are becoming less effective because of antimicrobial resistance, and the pharma industry has a very complicated profit motive. And there are a lot of genomes that have been sequenced over the last several years. If you just open any database, you will see that there are hundreds and thousands of genomes from all the kingdoms of life that have been sequenced. Now, what is the connection between genome sequencing and infectious diseases, right? So although these quotes have been taken from an article written in 2008, it was written by someone who worked at GlaxoSmithKline, it's very relevant even today in 2023. So what David Pomplano says is that, to his surprise, although at that time in 2008, 650 bacterial genomes had been sequenced, they thought it was a dismal failure because they were unable to discover new classes of antibiotics. And so he basically concludes that genomics is simply not a good paradigm for discovering new drugs or antibiotics. Now, what I would like to argue, and I hope I can convince you through the next 2.5 hours or so, that it's not that genomics is a bad paradigm, but you need to have a way, you need to have a platform to understand how genomics impacts downstream factors in a cell. So it's not enough to just catalog all the genes. It's not enough to catalog just the proteins. It's not enough just to catalog the metabolites. But you need a platform that allows you for integration between all these molecular hierarchies. And only in that case, you will be able to use genomics as a good paradigm, because eventually it is the central dogma of molecular biology. It's the beginning, the inception of the central dogma of molecular biology. Another reason why you need to build metabolic models is, yes? Sure. So as we go through the talk, you will understand that, we start from a premise that the central dogma works from the DNA to RNA to protein, and each of these proteins, if they are enzymatic in nature, they would catalyze metabolic reactions. But it's not enough to just go in this direction to have this hierarchy, but you need to have a backward feedback loop as well. because it is the crosstalk between these molecular hierarchies. It's the integration of these, the interactions between these hierarchies that eventually define the cell phenotype. But you have to start from some point, and the best point to start is from 1953, from knowing what the DNA structure was, right? And to have developed the central dogma of molecular biology and, start your journey from there. But the journey is never complete, especially from a systems perspective. You need to not only look at the components, the reductionist view of what exists inside the cell, but then you need to integrate back to understand how these components talk to each other and how they eventually end up, with that emergent phenotype of that organism. Because it's just not enough. I mean, there have been so many methods in the last 1.5 to 2 decades, analytical techniques that have come up to measure genomes, to measure transcriptomes, to measure proteomes, to look at, so there are a lot of new omes that have been coined, right? I'm sure if you just Google omes, you'll find that there are resistomes and there are unicomes, and there are lots of omes that have come up. And why? Because measurement techniques have, have been discovered, and they've been able to look at the cell in a holistic perspective. But at the same time, what they do is they allow you to go through one segment, or as I like to say, the left side of biology, which is the reductionist component. And that is very critical. But eventually you need to go back to the right side, which is you have a bottom-up approach. And then you integrate all this information backwards to understand why circuits behave the way they behave, why properties, certain emergent properties come up. So, yeah. So another example is of why we need to build metabolic network models is that, we live in a world where petroleum is declining, right? So we need to develop more green processes. So we need to use more techniques like metabolic engineering to make compounds using biological processes. So what metabolic engineering does is it views the cell as a chemical factory. And so just like in a chemical factory, you have unit operations, separators, reactors, connectors, controllers. In a cell, you have these, you have organelles, and you have pathways. But the only difference, the major difference between a chemical factory and a microbial factory is that the level of sophistication needed is much higher in a cell as compared to in a regular chemical plant. And how do you understand, how can you fine-tune a small pathway that is of your interest to make a compound that you want to commercialize? For that purpose, it's necessary not just to, again, go through this list of omics data, but to figure out how do you integrate all this? How do you understand this emergent property of making this particular compound of interest? Because the cell did not evolve to satisfy your bioengineering objective, right? The cell evolved through Darwinian processes, and the cell basically, wants to maximize fitness and growth. So it really is not tuned to the way of thinking that we want it to sort of satisfy. And these are just some examples. So with this background, let me come to something very basic. I don't think, so how many of you have some sort of chemistry background or biochemistry background? Okay, so probably 5%. Well, that's good. So let's assume two things, but how many of you have a biotechnology background or a microbiology background? Okay, another 5% or 10%. So we have about 20% audience, right? And the rest are computer scientists or physicists or mathematicians, I'm assuming? Okay, all right. Okay, so let's so when I said that I am an emergent systems biologist, right? So what I do is I look at systems. Systems can be at different levels. Just a few minutes ago, I described this whole omic revolution where I look at the whole cell, right? But for a few minutes, let's track back and look at the cell, look at a small part of a cell or define our system, right? So here I have a very small system, a toy system, and I have this, all these letters put in here in capitals. These capital letters essentially are compounds, okay, chemical compounds, and these small letters here are rates, okay. These are the rates at which these reactions take place. So I have a reaction network happening in a toy cell. This oval over here represents a toy cell. And for example, this S to A could be something, if you're a biochemist or if you're a microbiologist or a biotechnologist, you must have studied glycolysis, right, at some point. Or maybe even if you were in high school, I don't know, does high school teach 11th or 12th standard probably, right? So you must have some understanding of this. So imagine that it is a reaction like glucose plus PEP giving glucose 6-phosphate plus pyruvate, right? So that's what I want you to imagine. So now if I take this representation, this visual representation of a network and write down reactions associated with it, it would be something like this, right? So I have my reaction 0, which is S naught going to S at a rate of R0. My next reaction is S going to A at a rate of R1. Then I have S going to B at a rate of R2, and A going to C and B going to D at these rates. What I would like you to note is that if we draw a boundary here where I have currently drawn, then S naught is outside that boundary and it needs to come in, it needs to cross the boundary. C and D, after they get transformed, come out of that boundary, right? So these are things that you need to note when you look at any network. Now, if I look at this reaction scheme, and if I want to write a mass balance or material balance around these 5 reactions, what is material balance, right? I'm just trying to understand what is the rate of change of each of these compounds over here with respect to time. I only want to understand that. So I basically write down ds naught by dt, and because it's being converted to S at a rate of R naught, I write that is equal to R naught, and because it's being consumed, the rate is in negative, right? It's quite simple even for the non-chemists. Then ds, right, by dt. So what is the rate of formation of S? So that is dependent on S naught. It's also dependent on A and B. Similarly, you can identify all these compounds here are dependent on either formation of those compounds or the depletion of those compounds. So if you bring that information all together in terms of the rates at which they are being formed or consumed, you can write what is called a mass balance or a material balance. And the chemical engineers love this. How many chemical engineers in the audience? Okay, all right. So they definitely love material balances. So then once you have these differential equations, right, this rate of change of compounds written in this manner, what you can do is you can make it more compact. So how do you do that? You just rearrange this equation so that the d by dt, which is common amongst all of these, comes out, and you multiply it by a vector, right? That vector essentially is S naught S, A, B, C, D. In this case, it can be all the compounds or metabolites in a network, right? So you put that information here, and then you decide that you look at this and you can remove all the rates, right? You can create a rate vector because it's all about these 5 rates, and then you can identify what is the matrix that you need to multiply it to satisfy this equation, right? And so if you want to then equate this vector to x bar, right? So this vector is essentially a vector of all concentrations. This vector here is a vector of all rates. So you essentially have d concentration by dt equal to some matrix S, right, multiplied by the reaction rate vector R or a velocity vector. So here you can look at this matrix and either you can put it in as is, or you can also take the transpose of this matrix and put it in. And when you create the transpose of this matrix, what you end up is you end up defining reaction stoichiometries as columns. So these column vectors represent reaction stoichiometries.
The previous slide, the degradation reaction, say DSDT, shouldn't it be minus R1 multiplied by S?
No, it's only conversion of it's only the how much of S naught gets converted to S. So we are only looking at the unidirectional flow from S naught to S. Then when S gets converted to A, there you account for S naught coming to S going to A, and S going to B.
The second equation down there in material balance, ds dt,
yeah.
The second term and the third terms, if it's just minus R1 and minus R2, when S becomes 0, you get these unphysical terms, no? Shouldn't it be minus R1 S?
No, so right now we do not consider that. We consider that it is just a function of the rates. We do not consider concentrations at this point.
I think it's basically rates are that rate multiplied by concentration is basically R1.
No, but that can't be the case because then R1 would be time dependent. Because S is time dependent. So this is not really, I'm not understanding these equations. Because if you just have minus R1 in the second equation, when S becomes 0, you still have a degradation term.
So essentially when you have your, so you're basically saying that this is multiplied by the concentration, right?
The second and third term. This term is 0th order, that's completely fine. But second and third term should be first order reactions. Because otherwise I don't see how it makes sense.
So here essentially we just look at what is the rate at which S is being converted to A, what is the rate at which S is being converted to B, and we assume that this rate of change of S is really so fast that eventually we apply a steady state hypothesis to this, and that equals a 0. So maybe we can talk about it. So essentially we end up with an equation, with a set of equations like this, and we applied the concept of pseudostate. And so what is the pseudostate hypothesis? The pseudostate hypothesis generally is applied to intercellular metabolites. It essentially means that there's a high turnover of metabolites. The concentrations of metabolite pools adjust to new levels despite a large change in the environment. So it doesn't matter. So we are not saying that there is no change in concentration, but all we are trying to say is that the rate at which it changes is really so fast that the cell adapts to it really quickly. So since pathway metabolites are generally at pseudo-steady state, when you draw that boundary, whatever happens inside this boundary is, which is basically here we are considering it to be the cell, is at steady state. And therefore, your S, A, and B can be equated to 0. And if you then go on and solve this set of equations, you end up with one part that's more of an ordinary differential set of equations, and then you also end up with this linear part.
In this regard, if I really form a boundary, when I'm defining my concentrations, what should be my volume?
So I think it's not in this particular slide, but we consider a constant volume system.
Like including cell and the surrounding?
Yes, so it is constant volume. So this holds true only under a constant volume system. Because yes, otherwise there will be changes for sure. So now, under what circumstances, right, is this, why do you need to do this? So one of the reasons why we need to do this is that when you look at a cell, right, it is very hard to estimate Although I told you that there are a lot of methods that have come up to measure a lot of quantities, it is very hard to measure the rates of a lot of reactions. It's very hard to measure the concentration. So one has to really choose what are the most important compounds that you want to measure inside the cell. Now you might end up measuring. So the reason why you need to do this kind of analysis is that you want to eventually identify what are the best quantities that you want to measure, and so then the experiments can be done accordingly. So here R0 is the substrate can be associated with a substrate rate, right? An uptake rate of a substrate. R3 and R4 are the rates of formation of an extracellular product. And if you look at the equations that we had here, right, the star has 3 equations and there are 5 unknowns over here. We do not know R0, R1, R2, R3 and R4. Therefore, if you have measurements of only 2 out of these 3 extracellular fluxes, the S0, the C, formation of C and formation of D, which are generally much easier to do experimentally, then you can easily, you can solve these equations and you can understand the other unknown rates. Similarly, if you measure, so this is when you measure R3 and R4, you can again rearrange it. And the solution to this can be easily obtained by using matrix inversion. Without using matrix inversion in this case, but when you have a much larger matrix, you probably definitely need to use matrix inversion because it becomes very complex. Now, if you were to measure only rate of formation of D and and the substrate uptake S, then if you were to rearrange it, you end up with something like this. And again, you can solve it using matrix algebra. So now the question is that what would you do if you had all the 3 extracellular measurements available, right? How will you use that extracellular measurement? So essentially, if you have a network like this, where glucose is being converted to pyruvate to lactate, maybe I'll backtrack a little. I will come back to this a little later. So essentially where I'm going with this is that that matrix that we formed, right? That matrix that we formed, that matrix that we formed, that was S, that was basically, if you looked at the column vector, they were the stoichiometric reaction coefficients. So Bruce Alberts back in 1952 essentially created this concept of having reaction vectors based on the stoichiometry. And if you look at the way the equation pans out, the reaction rate vector can be connected to the rate of concentration change using the matrix S. And so if you remember your high school algebra, every matrix A, which is multiplied to a vector X, produces a vector B, which is a linear transformation that maps your X to B. So the S matrix essentially acts as a linear transformation between the space of reaction activity and the derivatives of the concentration space. And all these transformations can therefore be defined by the dynamic mass balance equation that we talked about. So essentially here what is written is that this is your dynamic mass balance equation, essentially dx by dt, where x is the concentration vector of all the compounds that are present in your network. V is your reaction rate vector, which defines the rates of all the reactions in your network, and S is that stoichiometric matrix which is put together using the stoichiometric reaction vectors of every reaction that exists in that network. Now, essentially, this matrix can be considered as a mapping operation between this V, which is the rate vector, and this rate of concentration. So essentially, you end up with mapping the reaction space with the concentration space. So essentially, this is a graphical representation of a matrix, and you all know that matrix has 4 subspaces, and the row space, the column space, the null space, and the left null space essentially define the 4 spaces of the matrix. And when you have this, the fact that your S matrix is essentially a linear transformation between your rate vector and your dx by dt vector, essentially ends up showcasing that the your flux solution space is coded by your row space and your null space, and your concentration space by your column space and your left null space. So all these transformations can be defined by this steady state mass balance equation. If you remember what a linear transformation is, if a matrix X is a linear, matrix S is a linear transformation between 2 different vectors, it has to follow the additive property, and B, it has to follow the, if any vector multiplies it, it needs to be equivalent to 0. So therefore, if you look at the left null space, you see, if you look at the null space, that particular equation, when you have S dot V equal to dx by dt, and under steady state your dx by dt is equivalent to 0, essentially defines all the whole solution space that corresponds to the steady state. So essentially, of the 4 fundamental subspaces of the stoichiometric matrix, the row space, it contains all the dynamic flux states and the thermodynamic transitions that occur. The null space contains all the steady state solutions to the flux balance equations or to the reaction rate equations. The column space contains the time derivatives of the concentration resulting from that mapping of fluxes by S, and the left null space contains all your time invariance of the network. So that's how the matrix eventually allows one to probe a reaction network. Now, the spaces that have been studied very extensively by scientists over the last couple of decades is essentially the null space, which essentially contains all the steady state solutions to those equations. Now, one of the reasons is probably because a lot of chemical engineers defined this field, and since they were, they were much more in tune with steady state analysis, this one got a lot of prevalence. But for people who would like to do research in these areas, I think the left null space is the one that provides a lot of potential, and unexplored questions exist in this space. So have I really finished 38 minutes already? Okay, all right. So then I'll skip through this part and I'll come to how do you, so now how does this relate to biology, right? We've talked about an artificial chemical system within within an oval, right? That was our network. So how do you connect it to the actual biology? So essentially, if you have a genome here, which is represented here, and this genome, we assume that it's a toy genome again, and it has 3 open reading frames, the green, the red, and the orange lines. We assume that these open reading frames code for different products. It can code for a regulatory gene, that regulatory protein that can bind to the DNA and either activate or repress that DNA. It can code for a protein, the triangle and the square, which can come together to form a protein complex. And that protein complex can eventually catalyze a reaction, say an isomerization between A and B, right? So these are the component interactions that take place in one of our, in a toy genome that contains 3 open reading frames. So ideally you would call this, in terms of biology, you would call it a genome annotation, right? A primary genome annotation. So if you were to take this information and map it onto a matrix, right? So let's assume that, let's look at the first set over here of the green open reading frame. So the components in this part of the diagram are represented by this green DNA, which codes for this regulatory protein that binds to this purple or lilac protein, right, and forms the complex. In this case, your red open reading frame forms this triangle and this square, and which eventually form this complex. In the last part here, you have these two compounds, chemical compounds, that interconvert to each other. So essentially, you can define these interactions, either these regulatory interactions or these protein-protein interactions or metabolite interactions, in a formalism like a matrix. So if you start with the easiest part here, it is this isomerization reaction. A is being converted to B, and if it is being, the stoichiometric ratios here are 1:1. So the stoichiometric ratio are the elements of your vector over here that defines this interaction. If it is being consumed, it's negative. If it is being made, it's positive. If you look at the protein complex formation, you have the red triangle and your orange square as components, and your product is this complex of both of these geometric figures. Now again, the ratios that are given here, as the toy example, are 1:1, and it forms one protein complex. So you essentially put those elements into that reaction vector that defines this protein-protein interaction. If it is being consumed, it's negative in sign. If it is being produced, it's positive. And similarly, for the regulatory protein interaction, you again have your DNA and your open reading frame that it binds to, and the DNA protein complex. So you can clearly see here that even though you have a 3 open reading frame toy genome, you quickly increase the matrix. You can represent everything in the formalism of a matrix, all these interactions. And a 3 open reading frame genome translates into a 8:3 matrix, right? So it's already compounded. So imagine E. coli, which all of you know, if you probably So each column is essentially a separate reaction. It represents a reaction, and each row represents a component. Mm-hmm.
That were to add with some other molecule, will it fall in the second row? Like if it wasn't catalyzed in the system, but taking part in the substrate, will it fall in the second row?
If it was not forming this complex, you mean? Or if it forms the complex but doesn't take part in this reaction? So then you would have to write out another set of reactions for the protein-protein interaction. So that particular house kind of complex, say it reacts with something else, binds with something else, and causes some dissociation in a different way. So you will have to write out the equation stoichiometrically, and then you can fill in the elements of another column. It will be another column. So essentially, here, based on this toy genome, you have 8 components only. If you had more components, you would add more rows to this matrix. If you had more reactions that were taking place further downstream, you would have more columns. So for a genome like E. coli, which has 4,400 open reading frames, you can quickly imagine how large the matrices become. And if you think about the human genome, then there are 20,000 open reading frames that actually code, right? The rest is the non-coding part. So imagine how large your matrix would become. But because it's a matrix, you are amenable to convex analysis, to other principles of linear algebra that you can apply, and you can solve them. So it makes it easier for you to compute what the cell can do. Now, what do you do once you have these networks, right? Once you've created this matrix. So once you create these matrices, once you've created the network reconstruction, you essentially can apply what is called a constraints-based approach to model or to understand this network. And essentially what that says is that you start with, you assume that you cannot know what a cell can do, and essentially assume that the cell has an unbounded space of activity. From that unbounded space, you successively apply certain constraints. These constraints are the stoichiometric reaction coefficients that I talked about. You can use limited thermodynamics, like understanding the reversibility of reactions in the biological cell. You can look at enzyme capacity, or the Vmaxes, the maximum velocity that a lot of enzymes participate with. You can look at genetic regulation, you can look at concentrations, you can look at kinetics. Like we showed the 4 subspaces. If you analyze different subspaces, you can understand, you can use kinetics and concentrations as well to sort of probe these spaces. So eventually, when you start with this unbounded space, apply these constraints, you end up with a polytope that is bounded, and this now defines the behavior of your particular cell, whether it's a microbe or a mammalian cell, it doesn't matter at this point. So essentially, this whole philosophy of constraints-based modeling is captured in a quote by Sherlock Holmes in his book, A Study in Scarlet. So essentially what he says is, how often have I said to you, when you have eliminated the impossible, whatever remains, however improbable must be the truth. So essentially, when you have this unbounded space that a cell can sort of tap into or leverage to do multiple things, there are some things that are not feasible because of different constraints. So if you apply those constraints successively, you can end up with a space, a mathematical space that you can analyze to understand what the cell can actually do. Now, generally, how do you apply these constraints? How do you get these constraints? As long as you can do experiments on your cell, you will be able to find those constraints that can be applied. Now, it's generally much more easier said than done, because it's hard to run experiments and get this experimental data that can be used as constraints, and B, it's not trivial to incorporate these constraints in these models. As I will show you in the next couple of slides, there are different layers that exist in this model. So if you look at just metabolism, that is the outermost layer that I showed, that the matrix. But behind it, I also mentioned that you can have protein-protein interactions, you can have, you can have the transcript and so on and so forth, right? So how do you then incorporate, say, for example, transcriptomic data into this whole equation? Not in the solution space. In basically, how would you start, how would you incorporate the concept of gene expression into this whole framework, right? Where you can define gene-protein interaction, protein-protein interaction, you can define metabolite-metabolite interaction. So you create from just being a metabolic. So when I showed you that initial toy network, which was just a set of compounds, is just a metabolic network, right? It's a chemical network, it's a metabolic network. So there are methods that have been developed to probe systems when you only define them as metabolism. And then you can create a set of linear equations, a system of linear equations, and you can solve them, right? But does that really define biology? No, it doesn't, right? So for biology to be defined, even for that smaller network or system, you need to include in your representation the protein, right, that catalyzes that reaction. You need to incorporate the gene that codes for a transcript that will eventually translate. So you need to include. Yeah. No, but that's what I just told you, right? About how you would start from a genome. So how do you start from a genome, and how do you end up with a genome scale metabolic reconstruction, which is not just about metabolism. It includes genomic information, it includes transcriptomic information, It essentially connects every single open reading frame in your genome to every single transcript that it codes for and every single protein that it translates to. And if it is a metabolic protein, if it's an enzymatic protein, then the metabolic conversions that it codes for. So that is how you start from a very chemical engineering concept, right, or very chemistry-based concept of reaction kinetics, and you apply it to what happens in the, you can't consider your a cell as a bag of reactions, right? You can't. So see, eventually the rates, most of the reactions in the that take place in the body are going to be either unimolecular reactions or bimolecular reactions, and they always take place at room temperature and so on and so forth, right? So that is the rate equations basically form the basis of being able to connect your genome all the way to your chemical reactions. That's how you should look at it. So essentially, so you can essentially use, since it's conveniently a polytope that is defined by that solution space, you can essentially understand, how a cell behaves by applying linear algebra principles, by, using this approach. So essentially, from this solution space, you can go down to understanding that single green point which defines the phenotype of your organism. So the edges that you see here in this bounded polytope are termed as extreme pathways, which basically are biologically reasonable combinations of reactions that can be combined at different weights that can allow you to describe everything that can happen in a cell. So essentially you move from the previous bounded polytope to this. Now, coming to your question about how do you connect this to biology, right? So I like to generally show this slide to explain to people about that. So the first, the left-hand side here essentially describes those reactions, right? These are 10 reactions of glycolysis that probably all of you are familiar with. The first one is the hexokinase reaction, glucose plus ATP giving G6P plus ADP. And we know that, let's assume that this is from E. coli and that the gene that codes for it is GLK. Now, if you look at the corresponding vector that defines this particular reaction in this matrix, every row here represents the compounds or the metabolites that are present here, right? So to answer your question as well. So this column here represents this reaction hex 1. The only non-zero elements in this column will be those compounds that participate in the reaction. If those particular, like these compounds over here, like FDP, DHAP, etc., which are part of the rest of this set of reactions, do not participate in this reaction, they will all have a zero value, right? So essentially this column here represents the reaction vector, the stoichiometric reaction vector of this hex 1. Similarly, each column here represents one reaction in this glycolytic pathway. All these rows represents all the metabolites that are a part of the glycolytic pathway. Now, so you therefore can connect so this is basically the biochemistry and this is the mathematics, right, the matrix. Now, what is missing here is the gene information. How does this reaction actually take place? And that is where the biology comes in. So for example, here, if you look at enolase, right, which is basically a 2-phosphoglycerate converting to phosphoenol pyruvate, this one is a monogenic reaction. So in biology, there are different ways genes end up working together to form proteins, protein complexes, and in some cases you have isozymes, in some cases you have, yeah, so I already said that, you have protein complexes, right? So for example, succinate dehydrogenase is a compendium of SDHA, SDHB, SDHC, and SDHD. So these 4 individual transcripts have to form individually, and they come together to form a protein complex to catalyze the reaction of succinate dehydrogenase. So essentially here, you use Boolean logic to define these. So this would be a monogenic relationship where you have the gene, the protein, and the reaction defined. Herein you have an isozyme. So pyruvate kinase the reaction can take place in the presence of either PYKF or PYKA. So you write this into your model. So you define gene-protein reaction relationships so that this information is coded in. In the case of gap D, the reaction gap D, you can note that there's one gene that codes for a transcript gap A, and then there are these 2 open reading frames that form 2 transcripts that come together to form your gap C, and then they become isozymes. So essentially you can code in for all kinds of gene-protein reaction relationships using this method, and therefore you can easily tie in the biochemistry, the genetics with the mathematics. So this is for only glycolysis, right? So when you do it for pathway by pathway, you eventually end up in something like this, where every dot here would represent a metabolite, every line here or a link is the reaction that takes place. And this is essentially the process of metabolic network reconstruction. This is the outermost layer of your model. What you have in the back end is actually all of this. You have your open reading frame, you have your transcript, you have your protein, and you have your protein relationship with your metabolic reaction. So you have defined your central dogma in this manner using Boolean logic to represent this, and material balance or mass balance, mass action kinetics to represent your biochemistry. So so this basically just talks about the complexity that would exist. If you look at Recon 1, an older network that defined human metabolism, there were 98 pathways defined in there, which included 3,300 reactions approximately and around 2,700 compounds. So essentially in the matrix that defined this particular generic human metabolic network, you would have 2,712 rows and 3,311 or 3,311 columns. And you would have all the other back-end information tied in whatever form, the gene-protein reaction relationship based on the biology of that particular cell. One more thing to note is that I talked about E. coli earlier, and E. coli, in E. coli you have only one compartment, right? There's the cytosol, and then there's a membrane. So you can divide the membrane into a periplasmic space and an outer membrane, or you can just consider it as one membrane, depending on whether you want to coarse grain or fine grain your model. But in case of eukaryotic cells, in case of human cell, you have almost 7 compartments, right? You have the Golgi apparatus, you have the mitochondria. So all this information is coded in over here. So if you had a reaction that occurred both in the peroxisome as well as in the mitochondria, you would have 2 reactions, but the localization information would be incorporated in there. So these are essentially steady state models. So you can understand what is happening in a cell at one point in time. You can create multiple such cases, and then you can integrate across them to understand the time course. You can create kinetic models, but that's not the scope of this work as the way I'm describing it right now. Yeah, so I think this is just a summary slide that essentially shows that if you have a network like this, if you have a biochemical network, if you have an organism, you can create a network like this. And if you create a network like this, it can be represented as a stoichiometric matrix. And because it's represented as a stoichiometric matrix, then you have the tools of linear algebra that are available. So, so over the last few decades, there are many metabolic reconstructions that are available across all the kingdoms of life. And there are many applications of these metabolic reconstructions. You can understand gene essentiality, you can understand what is the, under what conditions a certain compound is being made. You can understand how metabolic flux distributes throughout the network. You can understand if you knock out a certain gene, what is the impact on different phenotypes that you're interested in. It could be growth phenotype, it could be a product formation phenotype, it could be an aggregate phenotype. Say you want to understand mitochondrial function and you want to delete or you want to see the impact of a cholesterol forming gene on mitochondrial metabolism. Then you could create an aggregate reaction that you want to, that defines your mitochondrial function, and you could go and study that as well. You could also see what is the impact if you, generally a big problem in biology or in biological cells is resource allocation. And generally how cells are known to function is they want to minimize the cost to the cell. So they want to minimize the resources that are being used to attain a certain phenotype. So you can create, create a scenario where you understand that as well. So one of the most common constraints-based methods that has been popularized is flux balance analysis. So what exactly is flux balance analysis, right? So as I mentioned earlier, you start with all the reactions in the cell, you define the stoichiometry, right? Then you develop your mass balance equations, and so you get S dot V equal to dC by dt. And then you apply the steady state, the pseudo steady state hypothesis. So S dot V equal to 0. And then essentially this mass balance therefore defines a system of linear equations. Now generally when you have these set of linear equations, the system of linear equations, it is an underdetermined system and there are fewer equations than unknowns. And because of that, you end up with infinite solutions. So in this case, what we do is we introduce the concept of optimization, and we basically subject to linear equality constraints so that there is only one relevant solution, which is either the highest or the lowest value. So in order to solve this system of linear equations, which is underdetermined, we need to introduce the concept of linear optimization, and we would end up with either a minimum value or a maximum value, because you can either maximize or minimize this particular function. So what kind of function would you use that would be biologically relevant? So one of the first functions that was used was growth, because as I mentioned a little while earlier, cells have evolved through Darwinian selection, right? And essentially they are known to maximize their fitness or their growth. So biological cells lend themselves very well to this kind of approach where we look at maximizing growth in this system. Now, is there any growth? Have you guys ever heard of any reaction that represents growth in a cell? No, right? You must have heard of amino acid synthesis pathway, peptidoglycan, lipopolysaccharide. You could have heard of very complex pathways, but you've never heard of a reaction that defines growth per se. So how do we incorporate growth into this kind of formalism, right? So using, so what we do is we assume that we create an artificial reaction. This artificial reaction is based though on biology. We take biomass of any cell, so we grow cells, we take the cells out, we understand the macromolecular composition of the cell, And in the macromolecular composition, you would have nucleotides, you would have proteins, you would have, lipids, and so on and so forth, right? Now, every macromolecule can then be broken down into its constituent molecules. Like, for example, nucleotides would be ATGC and U, of course. Amino acids, proteinogenic amino acids generally constitute proteins, the 20 proteins. So you would basically go and experimentally calculate how much of every amino acid do I need to make, one, say for example, 1 gram of biomass. And then I would create an artificial reaction, which essentially is a linear combination of the demand on every single metabolite that exists in the cell that eventually contributes to that biomass. So I might have a slide that discusses it. Yeah, it's something like this. So essentially, assuming that these are some of the compounds that are needed for growth, you essentially go and calculate how much of ATP is required, how much of G6P is required, how much of F6P. So this has been, these 14 metabolites here have been sort of understood as they are the basic minimum. So if you want to make macromolecules like proteins, nucleotides, etc., the basic minimum metabolites that need to be made in a cell are these 14 metabolites. So essentially, you would calculate what is the mole fraction of each of these metabolites that is required to make 1 gram of biomass. You would just create a linear combination of all of these metabolites, and then you would use that as the objective for your maximization, right? Because we said that we made this assumption that cells generally maximize their fitness or growth. So generally, there are more than 48 components that exist in this artificial, if you might want to call it, biomass equation. And essentially, they are fractions of these metabolites that are required to make a gram of biomass. Right, so flux balance analysis is one of the most popular methods, and I'm sure many of you have heard of it and even probably used it. Now, there are many other methods, constraints-based methods that have been developed, which include understanding optimal solutions, like I mentioned, flux balance analysis, there's flux variability analysis. Some other methods cover understanding flux dependencies, like robustness analysis, phase plane analysis. There are also methods that have been developed to understand all the allowable solutions that you can calculate in a particular cell or network under certain conditions. And these are like the extreme pathway analysis, elementary modes. These essentially tell you more about the topology of the network. And of course, flux sampling. You also have methods that have been developed to understand altering phenotypes. Like, for example, if you have a genetic mutation, if you have a genetic knockout, how does that impact the cell? You could look at how does it impact one parameter, like the growth or a certain component production, or you could also look at how does it impact all the allowable, solutions of that particular network. What are all the metabolic flux distributions that this cell can take on at any point in time, given that, one of its genes is gone, right? So that is something you can look at. Other things that you could look at is energy balance. So we've talked about flux balance. Similarly, you can look at energy balance. You can look at redox potential. You can look at the sum of all the fluxes in your system. So that essentially can proxy for the cost that the cell, wants to expend to, say, for example, make biomass or make a product of interest, right? So in that case, if you want to minimize the cost that the cell is expending, then you would do a minimization as opposed to a maximization. If you want to understand, what is the maximum, say, energy allocation possible, then you would use a maximization function. So there are many methods that are available. We will not go into all of them right now. Right, so similarly, when you set up these linear optimization problems, you know that there are certain bounds and balances that you can include in your problem, in the setup of your optimization problem. And it is, yeah, I don't know why this slide is a little bit messed up, but yeah, I think you'll get the point. But then this essentially showcases that by adding, say, genomic information or transcriptomic information or metabolomic or proteomic information, or for that matter, even fluxomic information, you can sort of go from, a certain larger solution space without this data types. And when you add these data types, you can shrink the cone and try to come to a more, realistic perspective of this, what the cell is actually going to do. 6-7 minutes, and this is before question answers or after questions? Okay, all right. So I'll just summarize what we've talked so far, right? So essentially, I think this could be a good summary slide of this. Yeah, I think this could be a good summary slide. So essentially, when you want to build, if you want to create, so we talked about why we need to build metabolic network models. Why do we need to go beyond just representing chemical reactions or biochemical reactions, right? The need for incorporating all the whole central dogma hierarchies into your model. And when you create these models, therefore, you require to use a lot of information. The information that goes in is whether the reaction is present or absent, whether the gene is present or absent, what kind of cofactors are being utilized, what is the stoichiometry of the reaction, what is the reversibility, what is the localization, how do you define biomass, right? So if I want to define biomass of E. coli, it's relatively easy. E. coli is a very well-studied organism. But if I want to represent the biomass of mammalian cell, right? That's going to be a much more complex problem. And you cannot equate E. coli biomass to a mammalian cell. I'm sure you'll all agree with that, right? And so once you have all this information in place, then you end up creating this metabolic model with the back end of the gene, protein, and reaction relationship. And then you run your simulations and then you predict. Now, this type of modeling is an iterative process. You predict something, say you want to predict growth in the presence of a certain substrate. And you predict, yes, the in silico representation of that organism allows the cell to use that substrate. If you go back to literature or you go to the lab and do this experiment, and if it does grow, that's great, right? You check mark that, you validated your model and you move on. But however, when it doesn't, that is where those failure modes are really important for both the modeler and for the, for new biological discoveries. So when you get these failure modes, you actually go and probe whether you can change anything over here. Have I made a mistake? Is there something else? Have I omitted something? And so on and so forth. And then you put that back into the model. And then you again simulate for that function that was a failure. And if it then matches the experimental data, then you know what incremental advance that your model has made. And if it if it doesn't, then you have to go back to the drawing board again. So this whole process of metabolic network reconstruction and constraints-based modeling is an iterative process. It gets stronger the more and more constraints that you design, and the constraints that you design depend upon the experimental data that you can collect for the system you want to model. But again, there is one conduit that is also very critical. That is, how do you then translate that experimental data into a constraint that can be incorporated into your model? So define your biological objective, define your mathematics, how will you represent the biological question mathematically, and then how do you put it in a computational fashion and then get to your final phenotype. So with that, I will end this lecture. I'm happy to take any questions online here or offline as well. Yes, so nowadays it's very, Yeah, so nowadays there are many automated methods for creating models. Back in the day, people would do it painstakingly, either gene by gene or metabolite or metabolic reaction by reaction or pathway by pathway. But nowadays, what you could do is, there's already a compendium of models, curated models that exist. And in that set of models, as I mentioned, they cover the whole three kingdoms of life, right? So if you get a new organism, the archaea, the prokaryotes and the eukaryotes, right? So the three kingdoms of life of the, in the animal, not the animal, right? Archaeas are not animals, but yeah, you get, so basically the three kingdoms of life. And so what you could do is say tomorrow you discover this organism X and you go and sequence it, right? You need that sequence to begin with. You can go and compare this sequence against the sequence of say 5 or 10 or 20 model organisms. And when you compare the sequence to say there's, you have your let's, for simplicity's sake, say that glycolysis is a pathway in your new organism. When you compare the gene sequences of all the genes associated with this pathway with 10 model organisms and identify that, hey, this new organism matches, for example, say, some say the mouse, for example, right? It matches the mouse model the best, the mouse sequence the best. Then you know that the pathway, all the pathway requirements are closer to mouse. So therefore your stoichiometry, your cofactors, your localization, reversibility, all of that information will come from your mouse model. But if you, in that same organism X, if you have a pathway for lipid metabolism, and you see that lipid metabolism matches, say, salmonella more than a mouse, although your glycolytic pathway matched mouse, then the reaction, stoichiometry, cofactors, reversibility, etc., that information will be pulled out from that particular model, the salmonella model. So basically you have a compendium of genome sequences, you have a compendium of models, right? So you have so depending on the sequence, you sort of have an understanding what kind of cofactors might be used, what kind of biochemical evidence might be there, and so on and so forth. The best, but even after you create that automated draft reconstruction, you need to then manually sort of curate it and make sure that things are put in correctly. Because without that, without manual curation, so nowadays, so back in the day when we didn't have all these methods, it would take about 2 years to make one organism's model of the size of about E. coli or so. So 4,400 open reading frames, 2 years to create these models. Nowadays, it takes probably about 6 months to create, and then you would curate as well. So 6 to 9 months, you could create a model of the size of E. coli that is accurate. And most of these models have accuracy of about 90% or so. But what is very interesting is not the fact that 90% accuracy exists for the models that have good experimental data, but for organisms that are less studied, and you put together these maps and your accuracy levels are only about 60 to 70%, even those kind of models can predict phenomena reasonably well. So, because see, this 60 to 70% accuracy is based on all the genes in your genome, right? Which may not all contribute to functions that you're looking for. So, as long as you have well curated functions, you can probe certain aspects of biology. So stoichiometry is the same whether you're here or on the moon, right? But the way the network connects and reorganizes can be different. It will be different, right? I'm adding something. So whatever, see, if you have a certain pathway, say this new compound X, say you want to make carotene. right? Carotene is made by carrot plant. So you take the genes for carotene pathway and you put it into, say, yeast, and you express the genes, okay? And you put this into the model also, and you run your yeast model, and you see how much carotene is being made, right? So when you add this module for carotene production into this existent yeast model, you will use the stoichiometry that you know that happens in the plant, right? And there will be certain side products that come out. Those side products, if they exist in yeast already, they will be siphoned off into those pathways. So there'll be more flux through those pathways. If there are new compounds that are produced that were never present before, right, then they will sort of go into a sink, right? You will have to identify them, and you have to make sure that there is some way they can go and they don't block, because one of the main tenets of this is that accumulation doesn't happen, right? That's the steady state hypothesis. What is made is consumed, so there is no accumulation. So that needs to be taken into account. One thing that's very interesting is that because you add this pathway, there could be drain on some other pathway that would then prevent either your product from being formed or your biomass from being formed. Because in problems like metabolic engineering, you need to make sure that the cell grows happily and also makes your product, right? It satisfies its Darwinian objective and it satisfies your bioengineering objective. So what they actually found when they added the carotene pathway to yeast, this was done by J. Kiesling many years ago, and they realized that the cells just wouldn't grow. And they did it for a while. And then when they created this model and they looked at it, they identified that folic acid was deficient in the strains where you added the carotene pathway. So then just supplementing folic acid completely changed the scenario, and you could make more carotene, you could make more biomass, and everybody was happy. Cell division, not directly. Yeah, not directly. But
Because, I would imagine, because ultimately the cell division rates are also controlled by the metabolic processes. That would be actually very helpful, because some of us have been discussing through this workshop that you change certain conditions and your cell cycle kinetics change. It would be nice to be able to have predictive models for that.
So I think if you could equate the cell division process to like we define this artificial biomass reaction, if we can define different cell division stages, and I think that might not be very difficult to do with the kind of measurement techniques that exist these days, by putting in labels and then microscopy, a combination of microscopy and labeling of the same sample in two parts. I think you might be able to get information. I think it's very interesting. Nobody has done it. Yeah, nobody has done it. Yeah. Yeah, because I think it's only the very obvious that has been done so far. And people have not really, so I think that's where now the research angle comes in, right? What is the problem you want to solve? Those are the new areas for exploration. People have not, so the common ones, like metabolic engineering, like looking at targets for, disease states for drugs, then looking at how things are changing during disease. This, I think, has been the areas that people have been focusing on. But more fundamental processes, I think, I don't think many people have looked at it, yeah.
Yeah, because there's a lot of interest, there's especially when it comes to thinking of cancer as an evolutionary process.
Sure.
There are certain mutations, and there is this idea that often when you have drug resistance via mutations, there is this thought that there is a cost to drug resistance in terms of proliferation rate.
Sure.
Right. So could one then, with these models, engineer mutations, etc., and see whether one could predict?
Yeah, I think that is definitely something, because it's a little more one-to-one correspondence as opposed to now me defining something as cell division. I think that would be more fundamentally challenging. Whereas if you could make certain knockouts and sort of create these aggregate reaction demands on the cell to define different states, different functional states of resistant cells, then I think that's something that can definitely be done. Yeah. Yeah, so I'll talk about a little, not about I'll talk about mutations, I'll talk about rates, but in mammalian cells, it's a little problematic because there's a lot of maintenance, right, in a mammalian cell. A mammalian cell is not cell autonomous for growth. B, since these are steady state models, some of the other assumptions are there is at least there's a single growth limiting metabolite a lot of times. Whereas in mammalian cells, there's glucose and glutamine always need to be used, and they are both growth limiting. And sometimes there are other amino acids that become growth limiting. So sometimes, these predictions that you make are not 100% accurate. But then again, there's a degree of accuracy that you can always label. So yeah, that's interesting. Maybe we should talk about it.
Any other questions? Okay, if not, let's thank Anu once again.